This is an introductory video about the plasma membrane, the basic structure. So plasma membrane is the structure that separates the cell cytoplasm from the external environment. The main role of the plasma membrane is the tight regulation of the entry and exit of the particles into and out of the cells. Then to identify the plasma membrane during the cell fractionation, we have certain enzymes called as marker enzymes. The marker enzymes that are used here are 5' nucleotidase and alkaline phosphatase. So marker enzymes are used in the application of plasma membrane identification during the cell fractionation. The plasma membrane is made up of carbohydrates, proteins, and lipids. The carbohydrates are present in the form of glycolipids and glycoproteins, whereas the proteins themselves are present in the form of peripheral proteins present at the edges of the membranes, and whereas throughout the membranes, if they extend, those are called as transmembrane proteins. And finally, the phospholipids, they exist in the form of phospholipids and cholesterol. The carbohydrates in the cell membrane exist either as glycolipids or glycoproteins. The monosaccharides included in the cell membrane are glucose, galactose, mannose, fucose, N-acetylglucosamine or N-acetylgalactosamine. Coming to the functions of the carbohydrates in the cell membrane, they help in cell surface protection. The glycolipids act as glycocalyx, a protective coat on the surface of the cell membrane, protecting the cell. Hence, cell protection is one of the functions of carbohydrates. Then, using tight junctions, desmosomes, and the cell to cell adhesion is happening because of these carbohydrates. Certain bacterial surface carbohydrates act as antigens detected by the body. Hence, these carbohydrates, they are also giving the antigenic properties. And because of ABO blood group antigens, for example, ABO blood group antigens, which are also made up of carbohydrates present on the cell membrane, help in the cell identification. And finally, receptors. Most of the receptors are glycoproteins. So glycoproteins are present on the cell surface membrane. Those are recognized and helpful in the cell signal transduction. Coming to the proteins in the cell membrane, they are attached on the surface of the cell membrane. There are two types of proteins in the cell membrane, peripheral proteins and the integral membrane proteins. Peripheral proteins are bound to the cell membrane via ionic and polar bonds. That is one type of bonding. And if they are bound by covalent linkages to the lipids such as glycosyl phosphatidylinositol, which is derived from phosphatidylinositol diphosphate, a phospholipid, then that is called as GPI anchoring way of anchoring the proteins. This way of anchoring provides strong and stable linkage between the proteins, peripheral proteins, and the lipid layers. And suppose if the proteins are attached to the palmitic acid or myristic acid, those are called palmitylation and myristylation. Those are also happening through covalent bond linkages. Then coming to the integral membrane proteins. These are present deeply in the lipid bilayer via the van der Waals forces. When these integral membrane proteins span over the entire bilipid layer, those are called as transmembrane proteins. These are the ones that can act as receptors or ion channels or membrane-based enzymes. Then the cell membranes in different cells have different compositions of carbohydrates, proteins, and lipids. For example, the plasma membrane of the liver cells has highest percentage of proteins, 54%, and least amount of carbohydrates, 7%. Whereas the myelin sheath cell membrane, it contains 75% of the lipids and 5% of carbohydrates. That is, the proportions of these phospholipids, lipids, proteins, carbohydrates, they vary from one cell type to another. Coming to the lipids in the cell membrane, there are three types: phospholipids, glycolipids, and cholesterol. Among the phospholipids, we have phosphatidylcholine, ethanolamine, serine, phosphatidylinositol, plasmalogens, and sphingomyelin. Whereas glycolipids are cerebrosides, sulfatides, globosides, gangliosides. Next, coming to the phospholipids, every phospholipid is having a hydrophilic head, that is water loving head part of the phospholipid. Because of the phosphate groups, these are charged. Hence, the polar part of the phospholipid is usually the hydrophilic head. And these hydrophilic heads are always facing towards the aqueous environment. Always facing towards the aqueous environment. The other thing is hydrophobic tail. This is the tail part, but these are water hating and they do not contain any charge groups, so they are non-polar and they are always towards the inner side of the membrane away from the aqueous environment. Coming to the glycolipids, they are present on the surface of the plasma membrane, that is facing towards the outside part of the cell. Then cholesterol. The cholesterol can be present either in the free form or in the ester form, present in between the tails of the phospholipids. The ultimate function of these lipids in the cell membrane is basically acting as a permeability barrier and maintaining the fluidity of the membrane. So coming to the fluid mosaic model of the plasma membrane, the lipid bilayer. The first thought was plasma membrane was just a lipid bilayer, which was originally proposed by Davison and Danieli in 1935. Then later, the fluid mosaic model was proposed by Singer and Nicholson in 1972. According to this model, every membrane has two sides. One is the extracellular side, that is the facing outside, and the other is the cytoplasmic side, that is the inner side. And as you already know, it is made up of carbohydrates, phospholipids, and certain proteins. So among the phospholipids, as you can see shown by this arrow, these are the phosphate heads, the polar groups. And then you have the non-polar tails, the phospholipid tails. We will be focusing more on the phospholipids coming to the fluid mosaic model. So the phospholipid head and the phospholipid tail. Then coming to the bilipid layer, there are two layers of lipids. If this is considered as the first layer, the upper layer, this is the lower layer. Upper layer is towards the extracellular side and the lower layer is towards the cytoplasmic side. As you can see, the upper layer is mainly rich in the phospholipids containing choline, phosphatidylcholine, whereas the lower layer or inner layer is rich in ethanolamine or serine containing phospholipids, that is phosphatidylethanolamine or phosphatidylserine. Then each layer can be considered as a leaflet. So according to the fluid mosaic model, this head is made up of 1.1 nanometer thickness, whereas the tail is 1.5 nanometers in thickness. So each leaflet is roughly approximately 2.5 nanometers thickness. So 2.5 upper layer, 2.5 lower layer, 5 nanometers minimum thickness of membrane. And the middle gap we can consider that as a 5 to 8 nanometer thick plasma membrane in the cell. Then within the plasma membrane, the movement is only on the lateral side, from side to side. This free lateral movement is called the fluidity of the membrane. Hence the name fluid mosaic model. The movement is only in the hydrophobic core, lateral wise. Whereas the movement from top to bottom or from bottom to top, that is the flip-flop movement, is usually restricted. But in cases of apoptosis, this flip-flop movement is seen. So coming to the factors affecting the fluidity of the membrane. So what are the factors affecting the fluidity? The first one is the hydrocarbon chain length. If the hydrocarbon chain length is short, it is more fluid. That means more movement, more lateral movement is seen. If the hydrocarbon chain length is long, there is less fluidity, less movement is seen. The second factor is degree of unsaturation. In the cis fatty acids, there will be kinks or bends. Wherever these bends are present, there are more chances of better binding of the hormones. So more fluidity because of more movement, because of the bends there is more fluidity, more movement, so more number of structures can come and bind. So that is the advantage of cis fatty acids. Whereas trans fatty acids, they are straight, so they are less fluid, and because of this they are, because of the straight nature they are closely packed, and because of the closely packed nature they are less fluid. Then coming to the third factor, that is the polar head groups. In three ways these are affecting the upper layer containing the choline phospholipids. The polar heads of the choline phospholipids are usually larger in size. Because of that, they prevent the close packing of the tails, and because of that, there is a little gaps and ultimately increases the fluidity. Whereas the lower layer of the bilipid layer containing higher amounts of ethanolamine phospholipids, there the polar heads are smaller in size. Because of that, the heads and the layers are closely packed. Since they are closely packed, there will be less fluidity. Then coming to the next factor, that is the charge. If there is greater charge between the heads, we already know the phospholipid heads are the polar because of the phosphate. If the higher charge is present, the membrane becomes more stable. More stable membrane means less movement, so less fluidity. And finally, hydrogen bonding. You already know that between this fluid layer, lipid layer, the proteins are embedded or anchored. If that is strongly bonded, so there will be more stability to the membrane and there will be less fluidity. Then the next thing is content of cholesterol. If the cholesterol level is more in the plasma membrane, the fluidity of the outer layer is decreased. The outer layer fluidity will be decreased if cholesterol is increased. But on contrary, if the cholesterol levels are increased, the fluidity of the inner layer is increased. And wherever these cholesterol levels are implicated in certain diseases like spur cell anemia, alcoholic cirrhosis, lcat deficiency, alzheimer's, and hypertension. In all these cases there will be excess cholesterol, there will be decreased fluidity. Because of that, the receptors of the channels become non-responsible, so the cell will not take any action. No signal transduction will happen because the receptors are not functioning. Then coming to the next one, that is microdomains and caveolae. What are microdomains? We already know in the earlier part of the video, we have learned that GPI anchored proteins. Wherever these GPI anchored proteins are bound to the surface of the plasma membrane, those areas are called as lipid rafts or microdomains. Basically, these microdomain areas are rich in cholesterol and glycosphingolipids. And what are the functions of these lipid rafts? Endocytosis, G protein signaling, cell signaling, and binding of pathogens. And defective G protein signaling can result in paroxysmal nocturnal hemoglobinuria. That is about the microdomains or lipid rafts. Then next comes the caveolae. What are caveolae? These are also the areas present in the lipid rafts, but flask-shaped areas. As you can see, the depression, flask-shaped areas of the lipid rafts. They contain proteins called as caveolin. And here the function of caveolin, the caveolae, is endocytosis of cholesterol-rich lipoproteins forming caveosomes, or transcytosis, allowing the antibodies like IgA to travel through the cells. So the factors affecting the fluidity, first the hydrocarbon chain length, smaller chain length, more fluidity. The degree of saturation, the cis unsaturated fatty acids have more fluidity. Polar head groups, based on size. Larger the size, more fluidity. Higher the charge, less fluidity. And then cholesterol role. Increased cholesterol, less fluidity on the outer surface, more fluidity on the inner surface. Then microdomains, they help in the G protein signaling and the particle binding in endocytosis, whereas caveolae they help in endocytosis and also transcytosis. So these are defective, the fluidity will be affected. So these are the factors affecting the fluidity of the membrane. Then next, coming to the membrane asymmetry. The word asymmetry regarding the membrane is that there is differential composition of the lipids and the protein contents in the outer layer and the inner layer. Outer layer is the layer facing towards the extracellular side, whereas the inner layer is the towards the cytosolic side. So as you can see, the outer layer is rich in sphingomyelin and choline containing phospholipids. We have already seen this. So that is one differential layer. Then the below layer, the inner layer, the cytosolic side layer, it is rich in the phospholipids containing serine and ethanolamine. So this is how the lipid composition is varying between the two layers, causing membrane asymmetry. Now coming to the proteins. Regarding the protein content, the inner layer contains the C-terminal ends of the proteins, and the outer layer contains the N-terminal end of the proteins. Now, coming to the functions of the plasma membranes, we have five major functions. The first one is the transport of the molecules. So, by transporting different kinds of molecules, these plasma membrane helps in uh, various functions. The first one is controlling the cell volume by maintaining the electrolyte concentrations, by allowing certain electrolytes to go out and certain to come in. The fluid balance is maintained. Because of that, there is control of the cell size. That is one function due to transport of molecules. Then the second one is giving the excitable property to the cells. There are some inorganic ions like calcium and all those things. Those ions movement towards inside or outside is maintained by the selective permeability of the membrane. Because of this movement of these ions, there will be changes in the membrane potential, and hence the cell can be excited or the nerve potential can be initiated. That is one function. Then there will be secretion or exocytosis. Whenever the cell needs certain substances, it has to release. There is secretion, exocytosis. That is also happening because of the plasma membrane, one of the functions. Then phagocytosis, engulfment of large particles such as bacteria with the help of monocytes and neutrophils is also taken care by the plasma membrane. Then pinocytosis. Pinocytosis, there is a common understanding that it is only cell drinking, only water drinking related to. But along with that water, whatever the solute particles are present, intake of those things also happen in pinocytosis. You have to remember not just the liquid, but also the solutes present in the liquid is taken into the cell via pinocytosis. So via transport of molecules, secretion or taking in, or allowing just transport in and out. By transporting, these are the functions: controlling the cell size, membrane potential, excitable property, secretion, phagocytosis, and pinocytosis. Coming to the second function, cell to cell communication between two cells. Because of carbohydrates present in the cell membrane, they help in communication. Coming to the third function, we have transmembrane signal regulation. Some receptors and some proteins and enzymes act as receptors. These proteins and enzymes are present on the surface of the cell membrane. Example, G protein coupled receptor. The epinephrine is the substance binding to this receptor. Because of that, inside secondary messengers are activated, and because of secondary messengers, there will be a response from the cell. So, transmembrane signaling and regulation is also maintained. That is one of also one of the functions. Then coming to the fourth function, that is compartmentalization. Within the cell, there are cell organelles. Each cell organelle is separated from each other by membrane, because each cell organelle is already having a separate membrane. Within each membrane, each cell organelle is performing its own function in the form of having a compartment to each other. So because of this mitochondrial membrane, mitochondrial functions have remained here only. Because of this rough endoplasmic endoplasmic reticulum functions are within this membrane. Nuclear functions are within this nucleus only because of this nuclear envelope. So they're forming compartments. So compartmentalization is also one of the function. And finally, membrane modifications. There are four modifications of the membranes. One is near the cell junctions. Near the cell junctions, because the membranes are modified, they help in intracellular transport and adhesions, cell-cell communication, cell transport. In neural function, coming to the neuronal cells, myelin sheath, formation of myelin sheath is also one of the membrane modifications. If the myelin sheath is defective, it can result in a disease called as multiple sclerosis. Then coming to the formation of synaptosomes, we have already seen acetylcholine release, secretion, exocytosis. That is also because of a membrane modification. And synaptosome means it is the region containing both presynaptic and the postsynaptic regions. That is where the neurotransmitters or the molecules needed are secreted. And finally, the intestinal epithelial cells, where the surface are modified into microvilli, which can increase the area of the digestion and absorption. So summary of the functions is: by transporting of molecules, they control the cell size volume, excitable properties, secretion, phagocytosis, pinocytosis, cell communication, transmembrane signaling and regulation, compartmentalization, and by via membrane modifications, several other functions. Now, coming to the clinical aspects of the cell membrane, what happens if there are membrane disorders, membrane damages? The first one is the kinds of damages. If there is a mechanical injury, for example, the sickle cell disease, here the shape of the cells is getting affected. Smooth round cells, biconcave cells are getting converted into pointed sickle shaped cells. Because of that, the RBC's lifespan is reduced and various other clinical features are seen. So mechanical injury to the cell membrane, erythrocyte membrane gets damaged by the precipitates of deoxyhemoglobin. So because of that, the erythrocyte membrane is getting damaged, the shape of the RBCs is changed, and the lifespan of the RBCs is changed. So one is mechanical injury. Then chemical injury. Chemical injury. So in the chemical injury, the snake bites. The snake venom contains phospholipases. So even the phospholipids present in the cell membrane are broken down by the phospholipases enzymes. And these enzymes are plenty in the snake venom. Hence, if a snake bite happens, the phospholipases will break down the phospholipids and all the cell membranes get destroyed, and all the organelles, their functions, the contents will be released throughout the body, and you know all the clinical features. Then next comes the hypoxic injury. Hypoxic injury, that means there is low oxygen levels. Because of low oxygen levels, the ion channels and the sodium potassium ATPase will be not functioning properly. Because of that, the membrane, the cell will not be functioning. That is the hypoxic injury. Then coming to the mutations of the membrane proteins. There are certain uh, proteins in the cell membrane acting as receptors. For example, LDL receptor. Each protein is produced from a genes, right? Gene transcription, translation, and all those things. So if there is a mutation in the gene, the LDL receptors will not be functioning. Because of the not non-functioning LDL receptors, the LDL will not enter into the cell, and there will be accumulation of the LDL. Then autoantibodies to the membrane receptors. There, the membrane receptors are not functioning at all. Here, the membrane receptors are functioning, but the body is producing autoantibodies, antibodies against its own cells, blocking the receptors. Because of that, the substance, when released, when it is supposed to bind to the receptor is not able to bind to the neurotransmitter, and because of that there is no cell signaling and no proper cell stimulus cell signaling is not happening. Then next, now coming to the defective membrane lipid composition, we have seen already the membrane protein defects. Now membrane lipid composition, if there is an enzyme deficiency like glucocerebrosidase. In the Gaucher's disease, there is deficiency of the enzyme glucocerebrosidase. Because of that, there is accumulation of glucocerebrosides in the cell membrane. And we already know the cell membrane is rich in choline containing phospholipids or ethanolamine or serine containing phospholipids. If instead of those things, glucocerebrosides accumulated, there is altering in the lipid composition and there will be problematic. And then now coming to the disorders of the membrane skeleton, membrane protein defects again. So in diseases like Duchenne muscular dystrophy and hereditary spherocytosis or elliptocytosis, there are defects in the membrane proteins. In DMD, there is absence of the protein completely. Dystrophin is the protein that is completely absent in Duchenne muscular dystrophy. And in hereditary spherocytosis, there is a defect in the spectrin or anchorin protein, resulting in hereditary spherocytosis or elliptocytosis. So these are the clinical applications of the plasma membrane. There is seven disorders, seven types of disorders. Now coming to the final part of the plasma membrane about the specialized membrane structures. First one is tight junctions. Tight junctions, as many people can think that tight junctions cannot allow transfer of any kinds of molecules, but that's not true. They can permit small molecules and calcium ions to pass from one cell to another because they contain hydrophilic pores which are very narrow, and the proteins present in the tight junctions are occludin, claudins, and cadherins. In cancer cells, there are no tight junctions. Then coming to the second modification, that is the myelin sheath. How is myelin sheath formed? There are Schwann cells in the nervous system. The Schwann cells contain Schwann cells membranes. All these membranes condense together, spiral around the axon, forming the myelin sheath. The components of the myelin are sphingomyelin, cholesterol, and cerebroside. So those are the components of myelin. And in between these condensations, there are some regions where the myelin sheets thin out. Those are called as nodes of Ranvier. Here, the nerve impulse jumps from one node to the another. That is how the nerve impulse transmits fastly. And in diseases like multiple sclerosis, there will be demyelination at discrete areas which will reduce the nerve impulse velocity, resulting in motor deficits and sensory deficits. Coming to the third modification, that is microvilli. The intestinal epithelial cells surface of the plasma membrane are modified into thin elongated structures called microvilli. They help in increasing the absorption of the nutrients in the intestines. Then macrophages also have similar thing, but they are called as pseudopodia. The membrane evaginates due to the fluid natures. The next one is the cytoskeleton. The cytoskeleton is also a modification where it is useful for maintaining the cell structure. The proteins present in the cytoskeleton We can classify them as microfilaments and microtubules. The types of microfilaments are spectrin and ankyrin, whereas microtubules we have tubulin. Functions of cytoskeleton are cell shape maintenance, cell motility, and chromosomal movements during the division, cell division. Then acting as molecular motors. As you can see, the actin myosin, the skeletal muscle contraction, these proteins they move because of the ATP, the chemical energy getting converted to mechanical work. The mechanical work is actin myosin get closer and there will be skeletal muscle contraction because of conversion of this chemical energy. The best example of molecular motors is ATP synthase. So these are the specialized membrane structures. So all the best. Thank you.